My name is Zach Chandler, and I'm going to cover sickle cell trait, the HbS allele. I'm going to discuss its origin, how it came about to spread due to its heterozygous advantage with its genetic changes, and how inheriting two of the HbSS alleles will cause genetic changes in the body that result in aggregating hemoglobin and cause sickle cell anemia. So in this video, the questions that will be answered is going to cover the HbS allele's origin. We'll cover when it occurred, where it occurred, what happened genetically, and why it became so common afterwards. Part of those answers will lie in where this genetic change is located. It causes an amino acid change, and the substitution in amino acids has consequences. The HV stands for the hemoglobin protein, and AS is the alleles, A is the average adult one, and S is the mutated one. And we'll discuss what happens when you have this condition, or what happens when you inherit both conditions of the HbS and another HbS allele. And to help explain these, I'm going to cover the inheritance patterns. I'm going to show some pedigrees and go through them. And then also I'll cover HbS's effect on sickle cell disease in the world today, which has definitely changed over a long period of time since this first HbS allele originated. So shown on the left here is a pedigree of the HbS allele's origin. As we see here, we have two wild-type parents represented in the white color with HbA alleles, and they have a child that has a HbAS allele. And this is actually strange because usually genetic information comes from half of the mother and half of the father, but sometimes mutations do occur. And this actually happened around 7,300 years ago in Africa, and we might guess it was in this area that's darkest here. And what happened was this child was born with the HbS allele, and due to its heterozygous advantage, it would spread worldwide given resistance, which was necessary in a time where medication was not available, but malaria would kill many people in this region. So this individual would have the sickle cell trait, and he would be able to pass it on to his children, so they would have an advantage. But over time, inheritance of this HbSS allele would result in sickle cell anemia. So here's an example of sickle cell anemia. It is an inherited red blood cell disorder. We can see a red blood cell here on the right. And this is a pleiotropic phenotype. As we saw from the GAG to the GTG, that single change, that one mutation in one gene causes multiple effects in multiple regions of the body. So having one mutation provides that resistance to malaria to a greater degree than people who don't have it, and vascular damage and the sickling of red blood cells could occur, but this is more rare whenever only one allele is inherited. So shown on the left is a pedigree of the HbS allele origin. This is an uncommon way of seeing a pedigree where you have two wild-type unaffected parents with these HbA alleles, and then the child has a mutated allele. So this would also be noticed as the first generation, which research points back 259 years ago, this individual was born. And with this mutation, it typically does not happen, but when it does, it's usually occurred when genes are shuffled through meiosis, and this can change physiology, which is seen when people are not getting infected by a disease, or in unfortunate cases when a disease is wreaking havoc on the body. But these variations provide an ability for natural selection to occur and evolution to change over time. So what is shown here is that the pink is the person is a carrier, and we can see that on this right side with a pedigree that is more normal and more easier to understand after this mutation happened. So let's say 259 generations later, there's these people that have this trait, and they have one wild type, and they have one HbS allele. that is affecting their beta chain of the hemoglobin within the red blood cell. So they can have children, they can all not know that any of them have the disease until those children have kids, and there's a 25% chance that there's no resistance to malaria, just a wild type like everyone else. There's a 50% chance that they will be carriers and have that advantage against malaria, but there's a 25% chance that the individual will get sickle cell anemia and will die of an early death, which is very common in Africa, where most of this actually occurs and goes unnoticed because genetic testing is not being done routinely and medication needed for those individuals is not being provided. So pedigrees are a bunch of circles and squares. The circles are females, the squares are males, and they allow us to simplify what's going on. But what's really happening is that you have an egg cell and a sperm, which are going to be haploid and have 23 chromosomes, not in a pair, being passed on to the offspring. And it's during the process of meiosis where these genes get shuffled that mutations can occur, and how we see things such as the HbS allele come into existence. So now I want to zoom in to where in the nucleus this occurs inside the cell. So if we could peer inside of this cell here and see inside of it, we would locate inside the nucleus that there are 23 chromosomes. 1 through 22 are the autosomal chromosomes, and you can see they go from larger to smaller structures, whereas the 23rd chromosome dictates the sex of the organism. So X comes from mother, and the Y comes from the father, or this other X here comes from the father to determine sex. And while these are all organized in these units here, they often can work in unexpected ways. So we might see chromosome 11 working with 16 or other combinations. So one example of that is 2 chromosomes make hemoglobin, or the HB proteins, and after 6 months of age, we start to produce adult hemoglobin, which can simply be abbreviated HbA. This contains 2 alpha chains and 2 beta chains, and this is individuals without mutations. So the 11th chromosome here will contain the HBB gene, and that makes 2 beta chains. So these brighter ones here we can see as 2 beta chains, and the chromosome 16 will contain the HbA gene to make 2 alpha chains of hemoglobin, which are shown darker in these regions. And this is a heterotetramer. It's hetero because you have beta and alpha chains coming together, and it's a tetramer because 2 plus 2 chains is going to be 4 chains total. And you have 4 heme groups here, which is where the oxygen will actually bind and be carried through the body from red blood cells. So the location of the HbS allele occurs in this region here. So you can see the darker region as a gene location where information is contained. This is chromosome 11. It's position P15.5. So P tells us it's on the short arm, whereas Q would have told us it's on the longer arm. And these would be the 2 options available for those alleles. And you have one position here, one position here. Shown on the left here, we can examine the HbA allele. This would be the normal wild type, where in the 6th codon region, GAG encodes for glutamic acid, which is what most people have. However, here on the right side, we can see the HbS allele, where we're having that mutation from GTG to a valine and having that amino acid substitution. So this would create a HbAs genotype, which would be a combination of both because the 11th chromosome shares these 2 different allele types. This would be known as a heterozygote because there's 2 different alleles being expressed, and this would be someone that has sickle cell trait as a carrier and would have the heterozygote advantage with malaria. So this slide shows the HbAs protein. So we have a hemoglobin here, and we can see if these are the brighter beta chains, that one is producing a glutamic acid, which is negative and wants to interact with the environment, and this is more typical, whereas the valine is the unlikely new mutation that occurs, and it is nonpolar, so it would not want to interact with the environment. This two amino acids being present from both alleles is what provides the heterozygous condition, and it is very unlikely that these hemoglobin would aggregate into rods except in extreme conditions, say if someone was dehydrated, exerting a lot of energy climbing a mountain, and oxygen levels were dropping the higher they went up. But this abnormal valine does provide a resistance against Plasmodium falciparum, malaria infections, so it's not that the individual will not get sick from the virus, it's just more likely they will not die from it and they'll recover so they can pass on their genes to their offspring. So the HbS allele enhances resistance against severe Plasmodium falciparum malaria infections. This is a protozoa that is carried and transmitted by female mosquitoes whenever they bite into blood and there's blood transfusion. It allows this protozoan to enter the body. It will go into the liver and start to affect the cells, and then it will eventually make it to red blood cells where the hemoglobin is located. But typically we have glutamic acid in the 6th position, and this change to a valine somehow affects its asexual reproduction. Because it's unfamiliar to it, it will be less likely to reproduce, thus the malaria infection will be lessened and that individual is more likely to survive and pass on their HbS alleles to their offspring, which will give them an advantage if inherited. So let's go into that. The HbSS, which can just be abbreviated HbS for a sickled hemoglobin protein, they have only the hydrophobic valines in the 6th position of the beta chain, and this will cause the HbS to aggregate into rods. So earlier we had a valine just here, but now we have a valine located on this side as well. And what that's going to cause is all of these hemoglobins are going to form in these long rods. And some really useful illustrations I found were from this book here, which is on sickle cell disease, and it shows this valine, and an artist portrays it as this is the one sticking out, and the chains from the other beta or alpha chains are in the 85th position are forming these interactions that cause the aggregation. And another thing to see here too is on the right side, the HbS molecules, whenever they are oxy and they have oxygen, they are not polymerizing and coming together. But whenever oxygen is removed, as is the case when oxygen is delivered to tissue and then it flows back through the veins back to the heart to be reoxygenated, this is where the hemoglobins begin to polymerize. And we can see these long rods here twisting around and around, and you can imagine how that would affect a normal biconcave red blood cell to form into its sickled shape. This is another good illustration as well from some of the same images, but this is more diagrams. I think this is also very good to look at. You can see these rods here as the hemoglobin are all forming in these long connected pieces. In here we can see the 6th valine. It's interacting with the phenylalanine and the leucine here in the 85th and 88th position. And it's these interactions over and over that are causing these rods to form. And then you could zoom in here too, and you could see this is where the mutation's causing this connection, whereas this glutamic acid was actually more helpful because it was pushing away and preventing these aggregations from occurring. So now that we've covered on a chemical level what is going on with the amino acids and what causes the hemoglobin aggregation, the next thing I want to focus on is how do these aggregated hemoglobins within red blood cells cause so many problems. And here we can see problems that people with sickle cell disease have to deal with through childhood, adolescence, and adulthood. Vaso-occlusions is the primary issue. This is because typically red blood cells are biconcave and they flow through blood vessels with little to no issues. But a problem that occurs is whenever these have strange shapes, they can start to block blood vessels and prevent red blood cells from carrying oxygen to that tissue, and this can result in issues. When it happens in the chest, it can result in acute chest syndrome, and it can be painful and hinder breathing. If it happens prolonged in certain organ regions, such as the spleen is a common one, where blood cells go through, if the spleen becomes blocked and there's no way for oxygenated blood to reach that organ, then it might start undergoing organ failure because it's not getting adequate oxygen to keep itself alive. This can also happen too whenever blood vessels to the brain are not providing adequate oxygen to the brain, and strokes can occur. And also if the pathways to feed the heart with blood so it has enough oxygen to pump, if that is seized through vaso-occlusions, then a heart attack can ensue and the person can die from it as well. So the inheritance patterns of this mutated HbS allele can be shown pretty well with pedigrees. It's an autosomal recessive, so inheriting that one allele, the HbS, will give you malaria resistance and a heterozygous advantage. So all these individuals here may have survived times where malaria was killing a lot of people, and they were fortunate enough to meet someone else and have children with them. And these people that met, they both are carriers of this allele, and the problem with that is 25% chance nothing will happen, 50% chance their child will still have that resistance, but there's a 25% chance now that they will have sickle cell anemia and they will die an early death if they do not get the proper medication or treatment needed to live with sickle cell disease. So in this conversation where we've talked about sickle cell anemia, we've covered that it's an inherited red blood cell disorder and it's pleiotropic phenotype. The HbS is one mutation coming from one parent, and having only this and the other as wild type provides a resistance to malaria. So the sickle cell trait has become a heterozygous advantage, and that is because the people who don't have any kind of resistance in a time before medication against malaria, they would just die and not be able to pass on their genes. And this is shown in multiple texts that I've seen, but this is a good image of getting at it. The HbS allele protects humans against malaria, and we can see that on the allele frequency on the left here and the incidence of malaria on the right, which match fairly well. So this sickle cell trait also follows an overdominance or a heterozygous advantage pattern, and it can be shown here on the left that this is a positive correlation. This could just be like being taller and stronger is an advantage. So if being taller and strong is over here on the right, then that would be fixed for the best allele for that location. But overdominance is whenever being a heterozygote is more advantageous, and it doesn't push all for gene A1 or for the allele A2. It is best to have A1 and A2, and that's what we see as we get this stable equilibrium, and both of these alleles maintain themselves. So you don't want to have HbAA and have no resistance to malaria, but then you also don't want to have HbSS and have sickle cell disease, which would be the lowest because you would die of a disease early in life. So the best would be to maintain the allele to keep the HbAS, as we would see 50% of the inheritance is going to be that if people are carriers of the trait and have children, then this variation would be maintained. However, whenever an individual has the HbSS, the two mutations from both parents, then we can see here with the two chromosomes, what occurs next is there is still the resistance to malaria, but that is less important because now we have enhanced disease blood cells that are definitely going to aggregate. And when these aggregate and form long rods, it damages the vascular system and can block blood flow, which is very detrimental and causes a lot of the effects and early death associated with sickle cell disease and sickle cell anemia. So homozygous sickle cell became sickle cell disease as shown here in red. And this is definitely the worst one to have because you would be affected and the disease would be very. There's also many types of sickle cell disease. There's over 50, and on the right in this table, you can see many different ones with descriptions to them. We've covered the HbS allele, which is very commonly seen in most of these over here. And we know that origin dated back around 7,000 years ago. So when the HbSS comes together, we will get sickle cell anemia, which is the most common and severe form of sickle cell disease. But there's also many other variations you can see in the same region, that beta 6. If a mutation goes from a glutamic acid to a lysine, then that will produce the HbC. So you can have HbSC genotypes, and this is actually the second most common sickle cell disease type in Africa. So here's a good overview of the HbS allele frequency shown in the top of this image. As we can see, and we've already covered with the origin of the allele happening in Africa, this allele has spread all around the world since then, and it's also shown in the bottom image here where malaria most often occurs. So you can imagine the areas where malaria is around people that can be infected, having this allele is an advantage. So it's more likely the allele would be more prosperous in these locations. So in the United States, there are 100,000 people that live with sickle cell disease. There's 1 to 3 million carriers that have the sickle cell trait but do not exhibit the sickle cell disease symptoms. And that is approximately 8 to 10% of the African American population, which I think is quite high. And worldwide, there are 300,000 sickle cell disease births annually, and most of those are heavily in Africa, and where these are occurring, people are not being genetically tested, and they're not being provided the medication and treatment to help people who are born with this disease. So as a result, many children are dying from not getting the help or the parents not knowing they're carriers. And it's understandable that there would be so many births if there are 100 million carriers worldwide with the sickle cell trait. And if they have no clue they have this trait, they would not know that they're putting their children at risk. So in summary, we covered the HbS allele's origin that occurred 7,300 years ago in sub-Saharan Africa. These genetic changes occurred on chromosome 11. In chromosome 11, you have a normal wild type HbA, but with this mutation, HbS allele, you have a heterozygous advantage against malaria, and this is why this HbS allele became so common in areas where malaria was more commonly killing people. So these people would pass on their genes, and they would receive this allele, and they would continue to have kids. But whenever two heterozygous people had a child with the HbSS, they would have sickle cell anemia, and we covered how the hemoglobin would aggregate into rods, and that would result in the signs and symptoms of sickle cell disease. Throughout this presentation, we've covered the inheritance patterns and looked at multiple pedigrees, and we've looked at the HbS effect at large on sickle cell disease, which is a group of 50 plus disease types, but all of them have a root in this origin that we've looked at with the HbS allele. Thank you for watching.